Hello friends, in the last video we discussed about peptidoglycan biosynthesis. The cell wall of bacteria is made up of peptidoglycan. In this video we are going to discuss about bacterial reproduction. So bacteria reproduces by two methods, asexual reproduction, second parasexual reproduction. Many places it is mentioned as sexual reproduction. This we will discuss in detail in next video. Here we are going to discuss about asexual reproduction. Reproduction is mostly asexually in bacteria because bacteria are unicellular. Among different asexual methods, most common is binary fission. So let us see binary fission method of asexual reproduction. It is the most common method of asexual reproduction. It occurs during favorable conditions which favors the growth or reproduction of bacteria. In that condition, this binary fission occurs. If we see in brief what happens in binary fission, one bacterial cell divides into two identical cells. So from one cell, two daughter cells are obtained. Now let us see detail of this binary fission. We will discuss about what are the steps involved in this binary fission. So first of all, bacterial cell grows, that is, it reaches its critical mass. Then replication of DNA takes place. DNA molecule duplicates. That is, from one DNA molecule, two DNA molecules are obtained. Both copies of DNA molecule bind to the cell membrane at distinct places. That is, these two copies are bind to different places in the cell membrane of bacterial cell. Then what happens? Bacterial cell elongates. It grows in length. As a result, distance between two DNA molecule increases. That is, DNA molecule segregate. So what happens when the cell of bacteria elongates? Two DNA molecules that are attached to two distinct places of the cell membrane, their distance also increases. That means these two DNA molecules move away from each other. Then a transverse septum forms at the midpoint of elongated cell. This results in its division into two identical daughter cells. So a transverse septum is formed at the middle of this elongated cell, as a result of which this elongated bacterial cell divides into two identical daughter cells. Then at the end of this process, two daughter cells separate from each other. Each daughter cell then grow and divide independently. This type of asexual reproduction is seen in E. coli and Salmonella and some other bacteria. To conclude this, let us discuss about some of the facts of binary fission. We have already discussed that it is most common method. It is rapid and efficient. Advantage of this is that bacteria multiply rapidly and increases their population, as a result of which bacteria are able to adapt changing environment. Because large number of offsprings are produced, these quickly colonize or inhabit in the changing environment. But it has certain drawbacks also. This rapid and efficient way of binary fission has certain drawbacks. Bacteria divide very quickly. That is, bacteria multiplies in every 20 to 30 minutes. As a result of which the foodstuffs get spoiled very easily, and certain bacterial infection or certain bacteria which causes certain disease are spread very quickly. Also, there is no recombination seen. Daughter cells are identical to their parent cell. This is because there is no recombination. Hence, there is no genetic diversity. Hence, their ability to adapt the new environment is also limited. This binary fission occurs in favorable condition. When the condition is unfavorable, it slows down. Keeping this in mind, The perishable food items are kept in the refrigerator. In the refrigerator, the temperature is less, which is unfavorable condition for the bacteria. Hence, the multiplication of bacteria slows down and food is preserved. That's why these are stored in the refrigerator. Now let us understand binary fission with the help of this diagram. This is rod-shaped bacillus bacteria. This is genetic material of bacteria, DNA. This is cell wall and this is cell membrane. During binary fission, first of all, DNA molecule replicate. So here there are two copies of DNA. These are attached to two distinct regions of cell membrane. Then elongation of the cell takes place, as a result of which the two DNA molecules move away from each other or segregate. Then starts the formation of the cross wall. Cell membrane invaginates. Later on, cross wall is formed completely. Then the two daughter cells separate. So from one bacterial cell, two daughter cells are produced. These are identical to each other. These separate, then grow and reproduce independently. Now the second method of asexual reproduction is by bacterial endospore. So when it is formed, endospores are formed under unfavorable conditions. Conditions which are not suitable for the survival of the bacterial cell. It is formed in all species of Bacillus and Clostridium bacteria. Now let us see how endospore is formed. We will see it in detail step by step. So during the unfavorable condition, what happens? Part of protoplast containing nuclear body stores food. Then it undergoes dehydration. After that, this protoplast shrink and form condensed mass known as endospore primordium. A wall is secreted around itself. So endospore primordium secretes a wall around itself. Later, more wall materials are deposited by protoplasm surrounding the endospore primordium. Therefore, endospore is formed. So, with this, the endospore formation is completed. It is surrounded by 3 layers and rest of the cell degenerates. Now let us see some of the important facts of endospore. Endospore contain a central protoplast and a core consisting of DNA, ribosomes, enzymes and transfer RNA. So it includes everything that is necessary for the formation of new bacterial cell. Position where it is formed. It is formed either in the center or near the cell wall. Now shape of the endospore. Each endospore may be either circular, ellipsoidal or semicircular. These endospores are metabolically inert and highly resistant structures. These can withstand or survive unsuitable temperatures, radiation, pH, drafts, antibiotics and chemicals. When dry, these can withstand heating at 100 degrees centigrade for several hours. So in the dry condition, when it is heated at 100 degrees centigrade for several hours, it can survive. Now germination. How and when endospores germinate? Under favorable conditions, that is when the favorable conditions return, endospore imbibe water. Then outer layers rupture. Endospores are dispersed and when these fall on a suitable medium, these germinate into new bacterial cell. So what we can conclude that endospore formation is a method of perennation, that is to tide over unsuitable or unfavorable conditions. As only endospore is formed in one bacterial cell, on germination it gives rise to one new bacterial cell. So in each bacterial cell only one endospore is formed. And when this endospore germinate during the favorable condition, each endospore give rise to only one new bacterial cell. Hence, what we can see that its number doesn't increase or the number remains same. So it is called as reproduction without multiplication. If we see this figure, this is the bacterial cell. Under unfavorable condition, part of the protoplasm along with the nuclear body shrinks. Then it is surrounded by 3-layered wall and rest of the cell degenerates. This endospore helps bacteria to survive during unfavorable condition. And when the favorable condition returns, then it germinates and give rise to new bacterial cell. So this is all for asexual reproduction. These are the two important methods of asexual reproduction in bacteria. In the next video, we will discuss about sexual reproduction in bacteria. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.